Now in this topic we are talking about the growth of the cell. So what happens when the cell grows? Yes, it increases in size and it increases in number. You are right. Now there are two reasons why cells divide. Now what happens is when the cell increases in size and in number, then there is a load on the information, and that information is in the DNA. Remember I told you the DNA is like a library. So like this, the information gets overloaded. So the one problem faced by the cell is information overload. The larger a cell gets, the more demand it places on its DNA. So in order to meet the cell's needs, what the cell does is now starts dividing. The second reason is the exchange of materials. Now we know that the cell needs nutrients, so it takes in nutrients and the wastes are eliminated. Eliminated means it's thrown out through the cell membrane. So the larger a cell's volume, what happens is the surface area becomes smaller. So you have to see the surface area to volume ratio. Surface area means the outside area of a cell, and volume means what is the inner contents of the cell. Underline the last two lines. The larger a cell gets, the harder it is for enough materials to move across its cell membrane. Cell division solves the information overload and the materials exchange problems. Right. Now we will talk about the DNA. So the DNA is made into packages. The packages of DNA are called chromosomes, and it is these chromosomes that holds the cell's genetic information. So there are two types of chromosome now, the prokaryotic cells. So the small tiny cells like bacteria, which are the prokaryotic cells, they contain one single strand of DNA. But the eukaryotic chromosomes, that is the eukaryotic cells, have chromosomes which are extremely organized structures. Now what do we mean by organized? Now here what happens is the DNA winds around proteins called histones. This structure forms the chromatin. The individual chromosomes look like the letter X. Remember the alphabet X. The alphabets from A to Z, and the alphabet X is similar to that of the structure of chromosome. So the chromosome makes the separation of DNA possible during cell division. It is because of the chromosome structure, which you can see in this picture. There is the sister chromatids and there is the centromere, that is the joining point of the two sister chromatids. So the chromosome splits at the centromere and the two sister chromatids get separated. This is what happens during cell division. Observe the picture. Look at the histone proteins around the DNA. Now for the exam you need to remember sister chromatids, centromere, the chromosome. Now sister chromatids, because the chromosome is duplicated. The chromosome makes its own copy. You remember we have discussed this in the previous class, that the chromosome or the DNA needs to make a copy so that when the cell divides, each daughter cell gets one copy of the chromosome. revise the previous slide and this slide before we go to the next. Right. Now we come to the cell cycle. So what is the cell cycle? So cell cycle includes the processes that are taking place during the cell growth and followed by the cell division. Now, asexual reproduction, which I mentioned to you earlier, remember? Asexual, A-S-E-X-U-A-L, asexual reproduction is the type of reproduction that leads to genetically identical cells. So cell cycle here consists of the interphase. Now the interphase has three parts to it. That is the G1 phase where the cell grows, the S phase where the cell replicates its DNA or makes a copy of its DNA, the G2 phase where the cell makes its cell parts. Now it is the M phase for the cell cycle that can be divided into two stages. M for mitosis or the division of the nucleus, and followed by the cytoplasm division phase, which is called the cytokinesis. So what you need to remember for your exam, the cell cycle, what does it do? It is a series of events in the growth and division of a cell. How is the prokaryotic cell cycle different from the eukaryotic cell cycle? Well, the prokaryotic cell cycle, the cell grows, duplicates its DNA and divides by pinching in the cell membrane. But the eukaryotic cell cycle has 4 stages. The first 3 are the interphase, made of the G1 phase, S phase and G2 phase. After this is the M phase. And this M phase, the cell divides in 2 stages, the mitosis and cytokinesis. Cytokinesis is the division of the cytoplasm. This follows mitosis, which is the division of the nucleus. How will you compare between the binary fission in prokaryotes and the mitosis in eukaryotes? Both of them are talking of cell division. So here the mitosis is the division of the nucleus in eukaryotic cells only, that is in higher plants and in animal cells. This mitosis occurs in 4 stages. You know the names? Prophase, Metaphase, Anaphase and Telophase. We will discuss in the next slide. So in prokaryotic cell cycle, you can see the binary fission. The binary fission is the name of the cell division stages that takes place in prokaryotes. Here there is a regular pattern of growth, DNA replication and cell division that takes place. Now once the chromosome is replicated, the 2 DNA molecules, they move towards the 2 sides of the cell. Fibers form between them, stretching the cell, and then the cell is pinched inward. It is very important for you to underline this word. In the second last line, the cell is pinched inward. Now this cell pinching inwards divides the cytoplasm and chromosome between the 2 newly formed cells. Binary fission also results here in the production of 2 genetically identical cells. The same is for mitosis, when mitosis results in the production of 2 genetically identical cells. But the difference is that mitosis takes place in eukaryotic cells and binary fission in prokaryotes. So, why are we discussing this topic of mitosis? How is it important in multicellular organisms' growth and repair process? So mitosis allows the production of new cells. How does it do this? It helps in growth, increases in size. So when the organism becomes bigger and the growth is stimulated because of the mitosis. The second function of mitosis is repair and replace the damaged cells in different tissues. So for example, if you get a cut in your knee and the cells of the skin, the blood, the bones, they all are involved in mitosis. So it is by mitosis that the repair takes place and new cells are produced and the damage is treated in different tissues. So what is this 4 phases that take place in mitosis? The first phase is prophase. Now it is very important to learn these points, so you can make a note. The first point, in prophase, a cell's genetic material condenses. The chromosome becomes short and thick. A spindle starts to form. Now this spindle, which I told you before, these are thread-like fibers type structures which help to attach to the centromere and split the chromosome. Also, the nuclear envelope starts disappearing. It starts breaking down. So you have to note the 3 points. The cell's genetic material or chromosome condenses. The word condenses is important. So the chromosome condenses, spindle starts to form, and the nuclear envelope breaks down. Second, the metaphase. Here, these chromosomes now line up at the middle of the cell. So I told you the letter M, middle of the cell, M, is same like metaphase M. It's easy for you to remember. So these chromosomes line up at the center of the cell and the spindle fibers from the sides connect to the centromeres. In anaphase now, these sister chromatids separate. So the centromere splits. So there is a splitting at the centromere. This is because of the spindle fibers that constrict or become shorter. So this creates a pull and the sister chromatids separate and move towards the centrioles. Centrioles are on either sides of the cell. Telophase. This is the 4th phase where the chromosomes begin to unwind and a nuclear envelope forms around it. Remember, the chromosomes had become condensed in prophase. So in telophase, the chromosomes are no longer required to be condensed, so they begin to unwind. They become longer and a nuclear envelope again starts to form. After telophase comes the stage of cytokinesis, where cytokinesis is the division of the cytoplasm, and this division of the cytoplasm is different in plant cells from animal cells. So in animal cells, the cell membrane draws in and pinches off. So again underline and make a note of the word pinches off. So the cell membrane draws in and pinches off in animal cells. However, in plant cells, a cell plate forms. Now this cell plate helps in dividing the plant cell so that now there are 2 new plant cells. A new cell wall forms on the cell plate and 2 new plant cells are formed. Look at the diagram. During prophase, here you can see the chromosome condenses. So this which I mentioned to you in the previous slide. And you can see the duplicated chromosomes become visible. So you can see the sister chromatids. Outside the nucleus, you can see the spindle forming in green. The centrioles is the place where the spindle fibers start to form. What is happening to the nuclear envelope? The nuclear envelope is starting to break down. The second is the metaphase. The second phase of mitosis or metaphase is shown in this diagram. So during metaphase, the chromosomes line up in the middle or the center of the cell. The spindle fibers now connect the centromere of each chromosome to the 2 poles of the spindle. Look at the arrangement of the chromosomes and see the spindle fibers. Third phase, it's a very important phase, the anaphase. Here the sister chromatids suddenly separate and begin to move apart. And once anaphase begins, each sister chromatid is now becoming an individual or a separate chromosome. During anaphase, the chromosomes separate and move along the spindle fibers to the opposite poles of the cell. Look at the picture at the side. You can see anaphase, individual chromosomes now moving to the opposite ends of the cell. Here, the following anaphase is telophase, the fourth phase. So what is happening here? The chromosomes which were condensed now become long and they start unwinding. So look at the nuclear envelopes reforming and look at the chromosomes which are now long and unwinded.